Thank you very much, Pedro, and thank you very much for the invitation to all of the organizers. It's always an excellent opportunity to present work that we are doing. So my name is Lionel Broche. I work at the University of Aberdeen in Scotland, and today I'm going to present the work that has been done by the team in Aberdeen on the development of fast field cycling methods for imaging. So I'm going to give an overview of the current status of our research in terms of methods and clinical results, and also the aim of what we are doing. So for those of you who do not know, Aberdeen has a long tradition of making scanners. It actually is the home of the first ever MRI scanners that has been used for clinical purposes. So you can see that on the left. That was in the 1980s. And you can see that there's been quite a lot of development, but the last scanner that we have at the moment, the latest scanner, which is on the right, is strangely similar to what happened in the 1980s. So there is a kind of a style that Aberdeen likes to give to its own scanners. Anyway, today what we are going to see with this presentation is basically what field cycling can bring to medical research, or at least what I believe that this can bring to medical research. How we do field cycling in Aberdeen, what are the current technologies that we use and their limitations, and also what the outcome of the latest outcome of clinical research. There have been quite a few things happening since the last presentation at an EU Relax meeting, so I'm going to give you an update of what's happening. So first of all, I'd like to talk about what FFC can bring to medicine. It's quite a different word, basically. Since I started working in FFC, I've seen that there are three different words that are trying to merge together in Aberdeen. The first one is about fast field cycling NMR, and it's one that most of you will be accustomed for. You know a lot of things about that, a lot more than I do for most of you. And we all know that from the seminars that have been happening so far, that it can bring a lot of information on material. Indeed, with FFC methods, you can probe molecular motions in a way that are quite unique and non-invasive. So it gives a lot of value to this technology. So you can determine residence time and behavior of water in different compartments, phases, coexistence, and things like this. So it can give a lot of information on materials, which are allowed to develop models and to validate models so that you can make science progress by refining models and refining the understanding of materials science, which is great. Now, this has been ongoing for actually quite a long time. The first experiment in field cycling dates back from 1950s. But what we try to do in Aberdeen is to bring that to imaging. This is a different word. Imaging is a different word because, well, there is a big gap with NMR. Of course, there is a gap in complexity, because what we want to study in imaging are biological tissues. And biological tissues are a lot more different than isolated proteins, which are already quite difficult to study. And here we deal with living tissues. It's not only much more rich in terms of proteins and interactions, but it's also a dynamical tissue. So at least if you study living tissues, you have a lot of things happening. You have blood circulating, you have respiratory, you have exchanges of water through membranes and so on. So you have a lot of dynamics there. And of course, there is also a gap in the technology. You spend a lot more power on an FFC imager and you have a lot more components to put. But at least the aims are so far quite similar. So what we are aiming to do is to progress in the understanding of tissues, biological tissues. And then you have a third word, which is the word of medical MRI. And this one is a completely different ballgame. In a medical MRI, what clinicians want is to have answers to clinical questions. And most of the time, this is about anatomy. Most of the time, the question that they need to answer needs anatomical answers. So they need fine details, they need good contrast, but fine details is actually very important, maybe more important than good contrast, at least to my experience of talking with radiographers. And to do that, radiographers actually and radiologists have guidelines to follow, guidelines that are well established and that are efficient and robust, so that they know that when they want to investigate a particular pathology, they have a series of sequences to run, and they get images that they know how to to interpret. They've been trained to interpret these particular images. So it's a lot more of a dictionary approach. So the point is not to do research at all there, it's to actually deal with the patient. And on top of that, you have a lot of differences in the level of integration. MRI systems are not standalone in a hospital. They are integrated into an entire data system, so PACS and so on, so that they can be compared with other modalities, so to make sure that the clinicians can correlate or can cross-analyze results to get a better diagnosis. And of course, that requires a lot of training, tight regulations around it, and so on. So it's a very regulated environment. Now, how is that important for us? It's important because of the research, medical research that happens because of this. Because medical people are interested in anatomy and in details, there's been a very strong push in since the beginning of MRI to higher fields, because at the beginning of MRI, at least, going to higher field was a very good way to increase image quality. And so you can see on the graphs here, so that's two different graphs to show the same thing. Basically, the progress in field's strength for MRI has been quite constant through the years. But in terms of hospital use, it reached 1.5 and 3 Tesla, and most of the scanners since the year 1990s and so on are actually at 1.5 and 3 Tesla. And that is important for us because, well, if you look at you can do a small study. Took me 5 minutes. You can go to PubMed and have a look at the number of publications. So if you type a word here for MRI 3T or MRI, just only MRI, you can have the number of publications per year over quite a few decades, and you can compare how many publications contain some words. And then you can make a small estimation, a coarse estimation of the ratio of publication that contains a particular word within the literature of MRI. In here, I tried four different keywords: 0.5 Tesla, 1.5, 3 Tesla, and 7 Tesla. It gives, I think, a feel of what happens in history. And you can see that in the 1990s, the low field scanners had a kind of interest, but as medical equipment started to be more and more reaching 1.5 and 3 Tesla, the literature dropped for low field systems, which means that basically most of the medical literature is for high field systems. When doctors are trained, or when radiographers are trained for MRIs, they have very good references for high field, but not for low field, because they don't have the equipment, and the equipment is very rare. It turns out that at 0.2 Tesla or below, there is hardly any clinical literature. So it's difficult to actually approach the field of MRI for a clinical MRI at low field. Would it be just for that? And there is a very nice review as well that appeared recently from Sarakani and Salame about also technical aspects of low field, and why low field is actually that hasn't reached medical applications, and what actually it could bring. Problem is that there is no ground, or the medical ground is not ready for that. So, and yet actually low field is gaining momentum. If you go for low field MRI, well, for research, same as what I've done before, there was an interest in the late 1990s. Some people tried to advocate the use of low field because there was better signal. Then nothing happened after 2000, and it's catching up slowly because actually there are a lot of things to gain over there that cannot be reached at very high fields. Now, going back to MRI, to FFC, sorry. FFC has been used in the 1990s to actually probe the kind of contrast that could be observed at low magnetic fields in the context of medical use. There are various groups that are that have worked on the topic. There are a few ones that are famous: Koenig and Brown, Rink and Al. And they actually showed that dispersion curves were exhibiting different behaviors depending on the type of disease. There are a few reviews on the topic, and there are actually some very good results. But the interest at the time was to try and find an optimal field to actually design an MRI scanner at a particular field to target a particular disease, which is an interesting approach. And I believe that this could be an interesting way to use field cycling still nowadays to try and see if, well, with the interest of low field coming back, if field cycling could answer these kind of questions more precisely. So we could actually probe pathologies quite nicely with a field cycling system, but we could also go further and use an FFC scanner in its own right as a source of contrasts in the dispersion profiles. So the dispersion itself could actually add to the contrast, and that's an approach that is followed by other groups, mostly on preclinical scanners. One in Paris, IR4M, there's another one in Graz, actually, that I know of, and there are others, I'm sure. And actually, this can give a lot of information. The problem is that you have a lot of more complexity if you want to investigate what happens in biological tissues and understand how dispersions can bring information to that. You need to build a framework, a theoretical framework, and at the moment, this is it is happening, but it's at a very early stage compared to what you can find in medicine. So it's interesting to see that well, there is a shift of FFC from physics and chemistry to more biology and medical research. So this is there's a big gap in here, basically, which I hope that we can start to bridge. So to summarize, what I think FFC imaging can bring to medicine is basically to bring answers about what field would be the most appropriate for particular pathologies if people want to develop low field systems, but also to better understand the pathologies. If we manage to get good theoretical models of tissues, we can start to get insight on the pathological processes happening in the tissues, and this would be very interesting in a variety of pathologies. It's actually surprising to see how little is known in various pathologies about what happens at the molecular level. And maybe in the future, but that would be in the long term, FFC could become an imaging platform in its own right, but this is certainly something that would happen in quite a long time, because it needs a lot of certification and a lot of knowledge before this can be adopted by clinicians. Anyway, so this is my view of what FFC can bring to medicine. Now I'm going to present a bit about the technology that we have in Aberdeen at the moment. Well, you may all be familiar with FFC relaxometers. And if you will go for the simplest scheme of an FFC relaxometer, it's something relatively simple. I'm sure Jenny will agree with me. You just need a console and a few amplifiers. You need to convert a signal into a current, send it into a magnet, and play a bit with radio frequency signals, and you have a relaxometer. Of course, the practice is always more difficult than theory. And for this kind of system, you have a lot of things to take into account to make sure that the system can cope with a lot of problems, temperature fluctuations, stability, and so on. But anyway, these systems use resistive magnets. And because they use resistive magnets, they need large currents that needs to be controlled quite accurately. Again, as I say, you need radio frequencies, and in the system that we have, which is a smart tracer, similar to what you see on the picture here, there is a temperature controller, which is also very interesting for material science. Now, if you want to scale that into an imager, you have a few major differences. First of all, the magnet size. Of course, in an imager, you need to fit a patient inside the magnet, so you need a large bore. And that increases dramatically the energy you need to deal with the magnet by 2 orders of magnitude, typically. You also need to add gradient coils in 3 directions. You need them to be quite fast if you want to have a good imaging capabilities. You need them to be strong as well if you want to have some resolution. You need to add shim coils to make sure the field is homogeneous. You need to make sure that the temporal stability is actually very accurate. It's actually much more important than for relaxometry because in an image, the phase of the signal is actually paramount for the quality of the image. And of course, you have to make sure that the patient comes out of the scanner in a state similar to what he was before coming in. So safety is a very big concern. The way we do that in Aberdeen is, well, to use a resistive scanner. At the moment, the latest scanner we have, the one we use for the study that I'm going to present next, is the one shown in here. It's a resistive magnet. You can see it in the middle. It's actually quite nice. Resistive magnets are quite compact and they can be switched off, which is quite nice. It can reach about 200 millitesla and theoretically can go to 2 microtesla or below. It would depend on the level of care that you take to control the environmental field. So with a resistive scanner, the lowest range of the field depends on how good the quality you can reach to control the ambient field that is responsible to the limitation at low fields. Anyway, you can also change the magnetic field at 10 Tesla per second. So that allows to probe low magnetic field quite quickly and avoid too low waste signal. We have a field computation coil that you can see here. It's a kind of squarish frame. It was done by a Gareth in our group. And we also have dynamic correction, which is something that was supposed to be done, but unfortunately, there was all these COVID problems, and this is on hold, unfortunately. Hopefully, that will be finished soon, now that the lab is back to operation slowly. Anyway, the biggest challenge with resistive magnet for imaging is, of course, the power supply. As I say, you have to use very powerful power supplies, typically 100 kilowatts. The one we use can reach 125 kilowatts when it's a full blast. And you need to have it very precisely controlled. So typically, when we reach 0.2 Tesla, we need 1900 amps. And when we ramp up and down, typically need 600 volts. So that's actually quite high power. And because of that, we cannot use the scanner in DC mode. We need a 50% duty ratio to make sure that it doesn't overheat. The way that we deal with image acquisition is typically to use Cartesian acquisitions. And we do one acquisition per one line of acquisition per cycle. So for those of you who are not familiar with MRI, we don't record an MRI image line by line, but we use the K-space, which is basically the Fourier transform of the image. And it's the Fourier transform of the image, which is recorded line by line. So we do one cycle of field cycling to record the first line, and then we repeat that n times if we want an n by n image. So typically, we need to repeat this pattern 60, 90 times, 100 times, and then we get our image. The trajectory is actually correct, accurate. We have problem with phase. I'm going to come back to that. And of course, it's a bit slow. It's we can only image one slice at the moment, one slice at a time for a volume. We can only probe 4 fields and 6 evaluation times in 35 minutes. It's difficult to go for longer than 35 minutes because we need to install the patients, get scout images, and so on. So typically, that's the kind of examination we have. And of course, as I said, we have a problem with temporal stability because the field that we have for each acquisition of the line is slightly different. And that's because the current is not exactly the same. We need, in order to avoid problems, we need a stability of below 1 ppm in the current stability and reproducibility shot by shot. At the moment, it's about 10 times bigger. So the image looks like this, as you can see, instead of having a nice dot, so this was a bottle phantom, it tends to actually blur like this. This is an image from a 0.5 Tesla. It's actually a system. It's actually a much more dramatic problem than what we see on our scanner. But it's what I presented on a paper that presents an algorithm that corrects for this. So the post-processing of images by field cycling can allow to correct for these phase fluctuations, and we can get clear images after some processing. And that works reasonably well. Another problem we have with field cycling is radio frequency range. RF on the MRI scanners are typically in the range of 100 megahertz or above, and that makes it relatively easy to perform certain tasks. In particular, you can get radio frequency amplifiers and preamplifiers in that range quite easily. And when you work between 1 and 10 megahertz, it's more difficult to get these kind of devices. And you have also a problem. For instance, when you make RF coils, so the elements that are used to make the image, one of the strategies to increase the or to decrease the scan time and increase the image quality is to use coil arrays. And coil arrays require to have protection circuits to make sure that they don't burn, or the electronics doesn't burn when the transmit pulse is being sent. And this electronics is typically, as you see in this figure here. So for those of you who are not accustomed with electronics, well, the loop here is the coil. It's an inductor. It resonates thanks to a capacitor here. So when you have an inductor and capacitor, it gives this frequency, this resonance frequency. You typically have a lambda over 4, so a quarter wave circuit. So to make sure that the preamplifier doesn't see a large voltage, but low voltage and high currents. And then if you want to protect the preamplifier, you can actually short circuit the coil by using what is called a pin diodes. And the pin diode is something that short circuits when it sees a current, a DC current. You have to bring this DC current from somewhere, and basically it comes from outside. But to avoid the radio frequency signal to travel down this line, there is something called an RF choke, which is just a large coil, a large solenoid. At high field, that works quite well because you don't need such a very strong solenoid in here to block our radio frequencies. When you go to low field, you need something big here, and this solenoid starts to couple with the coil itself and couple with the rest. So it starts to be a mess. So it's difficult to actually make coil arrays at low field for this. It's one of the reasons. There are others. But yeah, that's, you have to bear in mind that there are complexities at low field that actually don't appear at higher fields, which is a bit counterintuitive. Anyway, once you have the images, you need to analyze them. And with field cycling, you have you have one extra dimension compared to other MRI systems. Well, of course, you have 2 dimensions for the image itself. You have 1 dimension for the evolution time, if you want to have an estimation of the magnetization decay. 1 for the evolution field. That's the advantage of field cycling. Of course, if you want to image a volume, you would have another dimension. We cannot do that at the moment because of time, but hopefully we'll do that once we have a coil array that works correctly. You need 1 dimension for averaging if you want to repeat your measure. You need 1 dimension for the coil arrays if you have several coils, another one if you want to have multi-echoes, and so on. So you quickly have high dimension images. And therefore, you have quite a lot of redundancy in your data set. This is not something we're exploiting very well at the moment. At the moment, the procedure we have for image analysis is, well, we start with the correction algorithm that I showed earlier on, but after that we do filtering. TGB filtering is actually working reasonably well. At the moment, we do just image by image. Again, as there is some work done underway to try and take advantage of the redundancy of the images. Once the image is filtered, we try to cluster it, and this is actually taking advantage of the whole data set, so at all fields, which is actually working quite well. Then we try to make an estimation for the NMRD profiles. So for each cluster, we provide the dispersion and this dispersion profile. And then we compare the dispersion profile with what we know from NMR acquisitions. So in terms of analysis of NMRD profiles, we have 2 different approaches. We have the Aberdeen style approach, as I say here, which is typically something that we coded in fit-like. You can get the code in here. It uses an ad hoc model. We try to minimize signal loss, try to extract as much information as possible, but we lose the physical meaning of the model. Unfortunately, the code has been broken for some time. I still need to fix the code. It can be used in some cases, but there are a couple of known issues at the moment. And then there is another approach that we are actually using at the moment, thanks to a collaboration with a group in Austin, which is basically to import the data and normalize the dispersion curve. It's similar to a master curve approach in a way, and then use a model-free approach. So to try to go back to a model that gives some information about the physics of the system, at least, tries to, but may not be the best in terms of retaining all the information from the data. So the normalization procedure is actually quite interesting. Typically, as I say, it's similar to a master curve approach. And the advantage of it is that it allows comparing a dynamical process. And this is actually one of the strengths of field cycling. In MRI, when you operate at one field only, you can never be sure of certain parameter, like the water content, for instance. If you operate with field cycling, you can actually see the dispersion curve, and you can remove or at least normalize this kind of effect. So you can compare things that become comparable. But for that, you need to make sure that you use a good reference. And it so happens that at high fields, the contrasts, the T1 contrasts, tend to be to reduce a lot in MRI. So that's because of the dynamics becomes very similar in tissues. Dynamics responsible for relaxation, T1 relaxation. So we can actually, in most tissues, normalize at high field. It so happens that in fatty tissues, it doesn't work very well. But in most tissues, if you take the region beyond the 4 megahertz, they tend to overlap quite well. And that gives you some differences in terms of the mechanism responsible for contrast. I'm just going to see if there is a question in the chat. That's okay. So we use this protocol to study what FFC can bring in medicine. We've been using that for some time now. And we're lucky in Aberdeen to have a hospital very close to us, which has a long history of research with the university. So it's actually quite easy to approach clinicians over there. It's also convenient because patients are just 20 minutes away from our labs, which is great. So we can actually have them taken to us on wheelchairs if they have low mobility. That's what happens with the stroke patients, for instance. And that can be done with one of the research nurses from the hospital. We have, as I said, strong support from the clinicians. We have also admin support for the ethics side. So the NHS has offices dedicated to the ethical conduct of projects, and we can actually reach them easily. And we can also combine FFC with the examinations that are being done at the hospital. So typically we can compare with MRI exams, and we can compare with histology reports or pathological reports of the patients, which is actually quite good. And another thing which is good is we have access to tissue banks. It's a bit complicated sometimes, but when it works, actually it can give a lot of data. A lot of the work that I'm presenting here actually comes from the tissue bank. Now, if you want to organize clinical trials, here is a kind of a diagram of what point of access we have, at least in Aberdeen, and there could be for anyone who wants to try during the time that the patient is at the hospital. So when the patient is admitted, typically there is a series of tests that are being done, which are then discussed by the clinicians. Depending on the diagnosis, so diagnosis is then established, and depending on the diagnosis, there may be some treatment or surgery or both. And if there is surgery, you usually have a resection, so the tissues taken away from the surgery. And that leaves different points of access for studies. Typically at the time of test and biopsies, we may be able to access the biopsies. That's quite rare though, because usually biopsies are taken entirely for histological analysis. But we could access the biopsy sometimes at some point to do NMR because it's non-invasive. So we could probe the biopsy and then send it to pathology. So that's one possibility, but it's a bit difficult, and ethically you have to be careful. Another point of access is when the diagnosis is established. You can have the patient report, and in practice, this is usually when we recruit patients. So I put recruitment here on the side because patients may be recruited at different points. But before the diagnosis is established, it's a bit hazardous to recruit patients because you never know exactly what they have. So that's another reason why biopsies are difficult to get. Once the patient is under treatment, you can do an image before and after treatment if you want to compare the outcome. This is what we are about to do with cancer, for instance. And then, of course, you can do FFC imaging of the resection after surgery. So you can actually get the tissue from surgery. You can also have FFC-NMR from the histological samples. So typically, the surgical resection is sent to the pathology department, and then it is analyzed. You can, if you have a pathologist in the team, you can ask that person to provide some samples at particular places. So these are the different points where you can actually that we actually use to access tissues. The limitations are that well, resections from surgery are usually fixed in formaldehyde, and that changes the composition of the tissue, which is a bit of a problem, of course. Typically 1 day in 4% formaldehyde. Also a problem of timing, and of course the care, patient care is something which is quite timely, especially for stroke or particularly aggressive tumors. So finding a place here is a bit difficult. And because we don't know what to find, what to expect, we usually have to start with a study of tissues or tissue resections to find what fields we can access, because with field cycling MRI, we can only access 4 or 5 magnetic fields. So we have to make sure that we record images at fields that are relevant. So typically, a study starts by FFC-NMR, so taking the core of tissues because it's easy. And once we have good understanding of what to expect, we go for imaging. Now, with this approach, we've been scanning quite a lot of pathologies so far, but the two that have made it to imaging are stroke and cancer, breast cancer at the moment. Stroke is something that we started to investigate in 2017 as soon as the scanner started to be usable. When it started to operate, we had the chance to start this project with Mary Jo McLeod, and we have recruited quite a few patients so far, with some success and some failures, of course, because the scanner is quite narrow and it's a development scanner. But anyway, what we saw with this study is that FFC can provide, or MRI at low field can provide some biomarkers. Now, the first line here, that's a result I have presented in the previous meetings of COST. The first line here is different modalities from the hospital, so CT, diffusion-weighted imaging from MRI, and T2 from MRI as well at 3T. And you can, if you compare that with the similar slice on field cycling, you can see that well, at different fields, that for 4 different field strengths, the region that is affected, so which appears bright in DWI, shows up quite nicely at low fields, typically 21, 2, and 0.2 millitesla. At 200 millitesla, there was something, but it's a lot less clear. So some contrast appears there. And we have had quite a few scans since then. Again, the 4 that I'm showing here are things that I've reported before, and which showed some positive results when compared with CT. Typically, these are markers of, well, either hypoxia or from hemorrhagic tissues. Here I say hypoxic, but it could also be hemorrhagic. And again, all the contrast that you see, well, the images you see here are derived from a contrast below 50 millitesla. But the problem with this study is that of course, we cannot access tissue samples. There is no surgery in there. We tried to preserve the brain as much as possible. So this is still ongoing. We need to reprocess the data with the chain that I've explained above, saying that's underway. I hope we'll have results soon to present on that. Now we have new results on the field cycling results for cancer and breast cancer. And this stems from a study that I started in 2012 on the study of sarcoma and carcinoma. Well, in tumors actually are quite complex environments. You have the tumor itself, you have the necrotic core, you have region affected around it. And I wanted to see if FFC cycling could actually bring some information on that. Recently, there's been some new insights on what FFC cycling can do with in breast cancer. There are two papers that have appeared in the field from the group in Torino. Basically, the first one shows that there is contrast due to water exchange through the membrane. So what you see on the top graph in here, that's 3 different cell lines, and depending on the level of inhibition of membrane, you can see that the residence time of water measured from field cycling changes. So field cycling actually shows some results that are depend on transmembrane water exchanges. And the signal that you see here is actually showing that as a result of a two-exchange model, which takes into account this effect, and shows that most of the relaxation in cells, at least in cell cultures, is intracellular. So the cell inside of the cell seems to be a lot more relaxing than the cell extracellular environment. Now, these are actually interesting results, and they shed light on what's happened in 2012. At the time, I studied 10 mastectomies, so 10 samples of breast cancer and 10 samples of sarcoma, so musculoskeletal tumors. Musculoskeletal tumors appear in a lot more variety than breast tumors, as you can see here, which is interesting and challenging at the same time. And at the time, I probed the tumor and also the peritumoral regions. Now, on breast cancer, we could see a lot of variety in the dispersion profiles, depending on where the samples were taken. The samples from the breast tissues appeared very different from samples of tumors and peritumoral regions. And there were quadruple peaks as well in tumor and fibrous tissues. Now, at the time, I already showed that there was a strong correlation between the offset of the curve of the dispersion profile of tumors in breast tumors with the prognosis of the patient. So apparently something could relate it to the aggressiveness of the tumor, and that could actually be related to the water exchange across the membrane, because aggressive tumors tend to actually have more water exchange. The cells have a more active metabolism. But reprocessing the data, by reprocessing the data recently, thanks to the group in Holstein, we also showed that the dispersion profile, the type of dispersion profile, showed some groups. And this figure here shows all the dispersion profiles from the study for all the patients, but normalized according to the procedure that I showed before. And what is quite remarkable is that there are these groups of dispersion profile that appear in here. Here are named A, B, and C. This group C only has one type of tumor. It was a mucinous multifocal carcinoma, so something quite jelly-like. Group A and B contains a bit of a mix of the rest. There is no clear separation between ductal and lobular carcinoma. We don't know what are causing these groups, but it's actually interesting to see that they appear. And if you look at breast tissues, you could also see two different types of dispersion curves. Here, there may be different interpretation, different reasons for that. Well, there may be tissue remodeling in some of them, but it could also be mixed tissues. It could be that some of the tissue samples had some fibrous tissues as well. It could be an error of sampling, so it's not clear. Unfortunately, at this stage, I didn't have the means to analyze the samples further. But anyway, from this study, we decided to go for imaging. Now, the imaging part is still happening and quite an early stage, but we already have a few results. So typically, we are including patients with lesions that are large enough to be caught by field cycling. Initially 1 centimeter, but we are shifting to 1.5 centimeters because we can only take 1 slice, and we've missed a few patients because the slice was not positioned correctly, unfortunately. Anyway, we take grade 2 patients with variety of different type of tumors. The protocol is, as I say, we take, well, we have to, before we take a field cycling image, we have to take a scout. So we take a series of images on 3D, and we select the most likely slice where the tumor should be. The problem being that this scout is done at 200 millitesla, where the contrast is not optimal. So it's a bit of a guessing game sometimes, most of the time actually, especially when the tumors are small. In here, there was a large feature. So you can see one of the best examples in here. And then we can compare that to the pathological report. So at this stage, we have the scan from 3D, and we, well, even if the contrast is not always good, and sometimes we don't have the scan itself, but we have the size of the tumor and the location where it should be roughly. So we try to guess what we see at this stage. Then we select the slice, and we do our field cycling experiment. So once we collect the data, we do this cluster analysis, select the region of interest, do the fitting, and we got a dispersion curve that we can use to extract some features, the Colorectal peaks, amplitudes, the dispersion at particular fields, and so on. Now, this is something we've done some time ago with HER2 patients, and that's something I showed in the previous presentations, where we could see different dispersion curve from different regions. Now, we improved that quite significantly since its last results. And what you see here are results from patients. So this particular series of pictures are from a breast cancer, and you can see the corresponding dispersion curves on the upper right. So this is one particularly good result, where the region S1 and S2 corresponds to the region of suspected tumor and the contralateral control, and they appear differently on the dispersion curve. So that's rather encouraging in here. We had another patient showing something similar. We had patients that not showing any change. So it's quite, you have to bear in mind that these are the two best results we have, but we have a lot of negatives as well, unfortunately. But at least we have some positive. Now, we also did some studies on sarcoma. So the study that I presented before also had 10 patients with sarcomas. And following the same procedure of normalization, we showed that, well, first of all, the normalization procedures, the scaling factor from the normalization procedure also correlated with aggressivity, but not as clearly as for breast cancer. But we also showed that there were groups of tumors in sarcoma as well. In sarcoma, we found five groups that are clearly different, as you can see in here. A, B, C, D, E on this graph, with the highest dispersion here corresponding more or less to the samples with the stiffest tissues. Again, there was no measurement of stiffness done at the time, but the type of cancer corresponding to the patients in group A, B, C, D, and E tend to be in an order of stiffness, typically observed at pathology. We also see that the contrast again seemed to appear below 100 kilohertz, so it's a low field contrast typically. Same as before with stroke and with breast cancer. And it's difficult to interpret. Sarcoma appear in a variety of shapes. And if you look at under the microscope, you can see lots of different things in there. So it's not easy to interpret again, especially without all the histological findings. If you look at the tissues surrounding the sarcoma, most of them follow a given trend. You have other type of trends as well, but most of them are typical from muscles. So it's probably different type of muscle or different type of tissues, and mostly muscles probed in here during the study. Interestingly, we also had some results from colorectal cancer. And in colorectal cancer as well, you can see some contrast below 100 kilohertz. So we had a first study with 11 samples of fixed colorectal tumors, and another study more recently with 30 fresh tissues that actually should have been finished by now, but unfortunately, the proteomics is still being waiting. So for that study, we will have the composition of the tissues, which is a big help. But yeah, we'll see soon, hopefully. The interesting thing with that is that, again, the contrasts that we observe between the tissues, normal tissues, and the cancerous tissues appear below 100 kilohertz. It's not always there. Some samples showed very little change in that region, but the one that showed any change were always systematically in this region here. So that's there is something that seems to come back for different types of tumors. It's not always the case. There was this project called Relax that started in Grenoble by Hannah Laresh, which showed that the dispersion curves tend in the brain. So this one focused on the brain tissues, brain gliomas, and the dispersion curves tend to have a lot of different shapes. So it's a bit more complex in the brain. So it's a bit hazardous to generalize quickly. But anyway, so it's almost time to conclude here. Typically, what I showed here is that FFC MRI can actually provide some contrasts that are useful for medicine. For maybe that would be just for the understanding of tumors. It may not be directly relevant to clinics. I don't know. At that stage, it's actually not clear what happens. But at least there is some contrast, and it seems to be at low fields below 1 megahertz for stroke and below 1 kilohertz for cancer, for most cancers. Dispersion information could be useful as well. Maybe the type of the quantification of the dispersion could actually bring something. There could be other markers to exploit. But the problem is typically that technically there still are issues. We could scan patients, but we have some issues with reliability. Of course, it's a homemade scanner, but at least we have the first in vivo dispersion profiles, and that's actually it's quite an achievement. And of course, we have to keep in mind that this is explorative work at this stage. But we are actually moving towards a new scanner. So future plans, maybe, at least I hope, is to deal with the bottlenecks that we have at the moment. We experience problems in the technology. We need to improve the technology. We need more centers for research. Again, if we want to convince medical centers that there is something to do in here, we need to be more than one center. We need to have better access to patients. And of course, we need collaboration if we want to have more visibility. So we have a project at the moment to make a new scanner inside the hospital in Aberdeen to increase access to patients, and hopefully to deal with technology problems that we have, to have something, a scanner which would be reliable. Now, to increase the visibility and collaboration, I'm in the process of making this open source. And well, I hope that, well, I'm dealing at the moment with administrative issues, but it actually looks quite promising at the moment. Most of the administrative questions have been answered. And I started a Wikifactory website, Wikifactory site to actually put the blueprints and the details of the scanner. Now, at the moment, it's still private because I don't have all the Go signals from everyone involved. But once I will have this, I'm planning to put all the detail of the project in Wikifactory. You can see a link later. Just one word on open source hardware. Most people think that reading open source is just a one-way process, but actually it's a lot more than that. If you're interested in that, I strongly invite you to have a look. But basically, you can retain some. Actually, you do retain some rights and duties when you use open source license. You keep the ownership of what you've done. You can also have ask some some restrictions on the sharing depending on the license you use. But typically, it facilitates the partnership, facilitates disseminations, and it also improves recognition, which is very important for research. Now, I'm really advocating for open source hardware. I believe that this is a very good way to explore with field cycling, but people have to understand what it is. And one of the problems at the moment is that because it's so new, there are a lot of legal aspects, especially for clinical devices. That's one of the things I'm trying to uncover before I release everything in open source. If you're interested, I've been very happy for you to contact me and to discuss. Anyway, thank you very much for your attention, and thank you to all the people who contributed to these studies. There are many of them, so there's a list here, but there are a lot of them. The work that I've presented is not only mine, far from that. I have been working with my colleagues on this, so you can see their name here. Of course, David, but also Gareth, James, Nicholas, who's been working on the breast cancer results, and well, others as well. And also, I'd like to thank the partners who have been working with us over the last few years and have done an excellent job of trying to uncover what field cycling can do with us. Thank you very much.